Mitochondria is the second largest cell organelle which is present in a cell and plays a very vital role in the functioning of the cell. It consists of inner membrane, outer membrane. It means it has two membranes. Double outer membrane is or double covering is present in case of mitochondria as well as the chloroplast or the plastids. Now, in between the inner membrane and outer membrane, there is an intermembrane space. This space is very important because the formation of ATP actually takes place in this place, and there are various other reactions also taking place in the same area. Mitochondria has its own DNA, and this F0 F1 complexes are very important in formation of ATP. ATP synthesis is coordinated by allowing or by exchanging the hydrogen ions out of the cell intermembrane space of the mitochondria. The ribosomes are present for protein synthesis, and cristae are present in the mitochondria to increase the surface area of activity, mitochondrial activity or metabolic activity in the mitochondria. The discovery of mitochondria was done by Colliker in the voluntary muscles of flight in case of insects. Then after the occurrence, these organelles, they are present in the eukaryotic cells. They are not present in prokaryotes, and when they are present in the eukaryotic cells, they are only absent in the RBCs because RBCs they need to be they need less organelles present within them because they have a lifespan of just 120 days, and in those 120 days the function is to transport the oxygen molecules from one place to different parts of the body, from the lungs to different parts of the body. The shape of the mitochondria is discoidal, sausage, oval, fibrillar, and granular. The size varies from 2 to 6 micrometer in length to 0.5 micrometer in diameter. If we think about the number of mitochondria present in the cells, in case of yeast cell, it is just one. Even in case of most of the lower organisms like chlorella or trypanosoma or protozoans, we see that they are present in very less number. Whereas in case of flight muscles, the number is almost 5 lakhs. So, according to the usage of the energy, the number of mitochondria increases in different sites of an organism or different body parts of an organism. If we see the membrane space, the inner membrane, the outer membrane, those bulbous structures which are seen, they are the F0 F1 complexes which will be capable of forming the ATP molecules by exchanging the hydronium ions. If we see the ultrastructure of mitochondria, the outer membrane is smooth, whereas the inner membrane has presence of the cristae, which increases the surface area for cellular respiration. This cristae can be branched, unbranched, tubular, or filiform. They can be at times complete or incomplete as well, because they ultimately lead towards these F0 F1 complexes. The electron transport particle which forms the ATP has three parts: the head, stalk, and base. These are the dimensions. 100 angstrom, 35 angstrom is the diameter of the stalk, whereas 45 angstrom is the size of the base. Now each of the structure is of different type. Like F0 complex is rectangular structure which is embedded in the inner mitochondrial membrane. The stalk part is a long structure which has a head over it, and the head piece is a spherical unit. So this is all about the structure of the electron transfer particle or the ATP synthetase molecule. If we see the mitochondrial chambers, the mitochondrial chambers are again of two types. We have already an idea: outer or inner membranous chambers. It lies between two membranes and is filled with less dense medium which is formed of water, minerals, and enzymes. The inner membrane is inside the mitochondrial membrane and it is filled with much denser and granular mitochondrial matrix, which is the site for Krebs cycle. In the Krebs cycle, protein synthesis as well as lipid synthesis occurs. The mitochondria has its own ribosomes present within it, and the kind of ribosomes present in the mitochondria is 70 S ribosomes. Here S is small, please make it big. It should be 70 S. S is the sedimentation coefficient. Whenever we take any substance, we can centrifuge it. Most of the cellular content, how do we segregate it? There is a machine which is quite similar to the blender, and the cell is broken down with the help of or it is crushed, then after it is blended. After centrifugation is a process in which the entire cellular content is rotated at a very high speed, and the one which is heavier particles settle down, whereas the lighter particles, they come up. So in this by this method, we can segregate the different cellular organelles. And 70S is the sedimentation coefficient of the mitoribosomes present in the mitochondria. The mitochondria also consists of its own DNA, which is known as mitochondrial DNA, and it is dispersed in the cell. It is also said that it is said as well as proved that that we all get our mitochondria from our mothers, and there is no contribution from the sperm for the mitochondria. The riboflavin, calcium phosphate, magnesium ions, and manganese ions are present in the mitochondrial membranes, inner mitochondrial as well as intermitochondrial membranes. The reason why these ions are necessary, because they help in the process of, or they are the enzymes or cofactors which accelerate the rate of reaction in case of Krebs cycle as well as lipid synthesis or protein synthesis. If you see this entire structure, this structure is the structure of ATPase molecule by which we can form the ATP. The outer side of the inner mitochondrial membrane and inner membrane part is shown. In between it lies the F0 and F1 complex. This F0 F1 complex is the same base, stalk, and head portion. You can see how when two hydronium ions, they are expelled out from the outer side to inner membrane part, one ATP molecule is formed. So this is the idea for formation of ATP molecules. We'll study this in details when we study the Krebs cycle. The semi-autonomous nature of mitochondria means that it is capable of performing all its functions on its own. It doesn't require any other structure for its functioning. The entire cell's nucleus or any other part is not that important. So what are the different structures which are present in the mitochondria which makes it self-sufficient? The 70S ribosome is present in the mitochondria, which makes it self-sufficient for formation of protein, and the entire process of protein synthesis can be done with the help of the 70S ribosomes. The mitochondria also has the presence of its own DNA molecule, which is known as mitochondrial DNA, and it is capable of formation of mRNA, which is messenger RNA, and this messenger RNA can be used to replicate the content of DNA. The ATP molecules also provide energy. So the mitochondria can form some of the required proteins, um, and is at times dependent on the nuclear DNA and cytoplasmic ribosomes for some other activities. So that's why it is said to be semi-autonomous. It is not completely autonomous but semi-autonomous, because at times it requires the help of nucleus for its proper functioning. Again, the entire structure of oxysome can be seen, which has presence of base, stalk, and head, and this is only 0 F1 complex. Please remember that the base is the F0 complex, F1 is the head part. The entire structure is as such that in between there is presence of porine. Why porine molecules are present? These porine molecules will be allowing only specific required molecules to enter inside the inner mitochondrial membrane or intermitochondrial membrane. In the inner part, you can see the presence of cristae, respiratory chain, and intracrystal space. Oxysomes are the particles which are capable of formation of ATP, and they are also said as elementary particles. There is one more name given to it, and it is the Fernandez-Morgan subunits. So they are also Fernandez-Morgan subunit. This is also a name given to oxysome or elementary particle for electron synthesis. The various functions of mitochondria we are already aware of. One of it is the production of energy. This is the primary job or the primary activity which mitochondria performs. Other than that, it also performs various other activities. Second most important activity is thermiogenesis. The meaning of thermiogenesis is production of heat. So, for production of or maintenance of the body temperature is done with the help of energy. The temperature is raised, and that temperature can only be raised if ATP molecules are present. And the ATP molecules play a very vital role in increasing or maintenance of the 37-degree centigrade body temperature of human beings. The formation of yolk is also done with the help of Golgi apparatus as well as mitochondrial cloud. The yolk or the nucleus of the yolk part, it controls the entire process or the entire formation of yolk. The yolk as well as the egg is done with the help of the mitochondria. Energy is required, so this energy is provided by this process. The formation of yolk is known as vitellogenesis. Mitochondria is helpful in formation of spermatid, the middle part or the middle piece of the sperm, which is also known as neighbor kern, and it helps in the process of spermiogenesis, the formation of sperm. Mitochondria also plays a vital role in triggering various diseases if it is mutated. So, in case of various diseases like muscular spasm or muscular discoordination, liver disease, sometimes sight problem, vision problem, gastrointestinal problem, or neurological conditions, even kidney failure can be caused because of mitochondrial mutation. If mitochondria is mutated, then energy is not provided to different part of our body properly, and this can be a result for Parkinson's or autism or cardiac dysfunction or even heart failure at times. So, any individual who is suffering from any of these diseases, if we check the mitochondria of those individuals, we find that the mitochondria is mutated, and because of that, we can see various faulty measures which are there, or various faulty functioning of the body can be seen in those individuals. There are various similarities between the mitochondria and bacteria. It is also said that initially the mitochondria was a kind of bacteria which was free living, and later on, during the course of evolution, somehow it was engulfed by a eukaryotic organism, and after that it started surviving. Rather than being digested, it became an integral part of the organism, and due to mitosis, every time the cell divided, it also divided along with it, and that's why it also contains its own DNA. So thinking about all these factors, we can quite think that maybe once upon a time mitochondria was a bacterium, but later on, due to surviving in a eukaryotic cell, it became an integral part of it. And now, after several thousand years of evolution, still it remains completely semi independent in the individual's body, and it is functioning along with its own DNA. It is not dependent on the nucleus for its division. So, what are the different factors which makes, or different points which makes mitochondria quite similar to the bacteria? The presence of Cristae. Both the mitochondria as well as the bacterial membrane, they have presence of some enfoldings. These enfoldings are known as cristae in the mitochondria, whereas mesosomes in case of the bacterium. Both the mitochondria as well as the bacterial cell has presence of circular DNA, which is double stranded and is naked. It is just dispersed as such in the mitochondrial structure. Third point is about the multiplication or the division. Whenever the mitochondria it wants to divide itself, it is only capable of division by the process of binary fission, which is the same case which we see in the bacterium. Bacteria also divides by the process of binary fission. Both the bacterial as well as the mitochondria, both of them contain the 70S type of ribosome, which makes it capable for protein synthesis on its own. And if we consider all these factors at once, we can think that maybe there was a chance, or there has been a lot of evolution taking place in the human beings with the addition of mitochondria within them. And maybe because of the presence of mitochondria only, the human beings they have become such capable and self-sustaining, because we can produce our own energy with the help of this mitochondria. And the entire process of this mitochondrial activity, because of the mitochondrial membrane, is capable of making a independent functioning of the cells in case of larger animals.